150 base pairs. Perfect. Okay, now let's try to understand very clearly. So in the yesterday class, whenever I'm explaining you the replication process, now I have told to all of you very clearly, the eukaryotic DNA replication is always very closely related with what, friends? Always very closely related with the cell cycle. If every one of you very quickly recall my words, and you all people know very well, we generally use it to say that G1 phase is the preparatory phase for the S phase, and similarly we use it to say that Z2 phase is the preparatory phase for the M phase. That is what we use it to read. And you also remember in the yesterday class I told you the initiation of the eukaryotic DNA replication occurs in two steps. What is that? The first one is called as pre-replication complex formation. And the second step is, friends, the pre-replication complex has to be converted into the active replication complex. So this is what the process of initiation. But if every one of you very quickly recall, this pre-replicative complex formation and active replication complex formation will occur in two different phases. As we discussed that pre-replicative complex formation occur in the G1 phase, continue everyone. But friends, the activation will be taking place exclusively in the S phase. This is what we all people studied. And this is the last slide we have completed in the yesterday class. All of you please pay attention and try to understand what exactly I'm trying to say here. So what is the name of the protein that is involved in the replication process? Remember that in case of a prokaryotic organism, friends, the name of the protein that starts the DNA replication, that is what we can call the DNA. And what is the name of the protein that starts the DNA replication in the eukaryotic organisms? Name of the protein only we can call it ORC protein. So ORC protein will go and recognize this particular replicator region in the DNA. As we discussed very clearly, friends, the moment ORC recognizes the replicator, it is going to recruit either CDC6 or CDC18. Then later, friends, there is a recruitment of the CDT1. Then what I told to all of you, the ORC, CDC6 or 18 and CDT1 all together going to recruit which protein? All together going to recruit a protein called as a MCM complex. And what I told you very clearly, MCM is the protein which is generally acting like a helicase enzyme. I have also told to all the students very, very clearly that these all events, whatever I have explained to you right now, these all events are taking place where? In which phase? G1 phase. And in the G1 phase, even though helicase is ready, but the helicase won't be active. That's why the DNA replication or opening of the double-stranded DNA never ever takes place in the G1 phase. What we discussed, hope every student remember, if the enzyme helicase want to be activated, what I told to all of you very clearly, out of 6 subunit of the helicase, 5 subunits of the helicase has to be phosphorylated. If 5 subunits of the helicase want to be phosphorylated, friends, what does it require compulsory? It requires an important protein called as SCDK protein. Please don't get confused with that particular point. Now, look into this. Every one of you look into this. So, now the enzyme the protein called as SCDK. Why did I use the word SCDK, friends? I have used the word SCDK because the protein CDK is getting activated only in the S phase. That's why we call it as SCDK. What it is doing, you only look into this. Out of 6 subunits, friends, it is phosphorylating at least 5 subunits. The moment helicase enzyme got phosphorylated, every one of you remember, it starts opening the double-stranded DNA. You got that particular point very clearly. Now, what's the most interesting point that I want you to understand here? Listen to me. Remember my words. So now, there is an important enzyme. I hope you all people remember, right? DNA polymerase alpha. In the last class, we all people studied very clearly. DNA polymerase is the enzyme which will add the primer. At the same time, friends, DNA polymerase enzyme alpha only add the nucleotides, but we all studied very clearly that it can add up to 20 to 50 nucleotide. What is the reason? The reason is that because of the low processivity. Then you all people know very well the polymerase alpha can be replaced by the delta. So you can see the picture very clearly here. This is the polymerase alpha. Sorry, the polymerase alpha added the primer. The primer is the orange color one. So after addition of the primer, polymerase alpha itself adding the nucleotides on a continuous manner. You can see that particular point. It is adding on a continuous manner up to 20 to 50 nucleotides. Later it is replaced by delta. Okay. My simple question to all of you, I hope you all remember. What is the name of the protein? Or what is the name of the substance or a protein which can replace polymerase alpha with polymerase delta? My question is, what is the name of the protein that is involved in the replacement of the polymerase alpha with the polymerase delta? Who will do that? RPA will do it. Maheshwari, no dear. Okay. PCNA, proliferating cell nuclear antigen. Okay, good. No. RPA function is what here? RPA will load the PCNA into the pol delta. Then the pol delta will start working, very fast with the highest efficiency. Here the replacement can be done by pol alpha to pol delta by the PCNA. Don't get confused. Okay, last and final question. I hope every one of you remember. What is the name of the regulatory mechanism in the prokaryotic DNA replication? Every one of you. What is the name of the regulatory mechanism in the prokaryotic DNA replication? Very good. What do we call it as? We generally call it as the sequestration. Sequestration is the name of the mechanism where the prokaryotic DNA replication is regulated. I hope you all remember. And similarly, friends, in case of a eukaryotic organism, how does the process of initiation is controlled? I can tell to all of you very clearly, the entire process of the regulation of eukaryotic replication is under the control of a SCDK molecule only. What I have told you very clearly, friends, the process of initiation in case of eukaryotes will occur in two steps. One is called pre-replicative complex formation, and friends, second step is activation of the replication complex. Correct na? Now the question is very, very clear. When the pre-replicative complex formation is happening, this is the most important step. Pre-replication complex will happen whenever the activity of the CDK is extremely low. So try to understand my words very correctly. Whenever the level of the CDK is low, look into the point. Entire pre-replication complex is forming. There is no doubt about it. But what is the point I want you to understand? I want you to understand a point that whatever the pre-replicative complex formation occur, but it is not getting active. That is the question. Pre-replication complex just formed, but it never be active. What is the reason? Why it won't be active? If it wants to be active, it requires a high concentration of the CDK. But here what we have written, the CDK activity is low. So what is my conclusion here? When the CDK activity is low, pre-replication complex will form, which means all the enzymes will assemble, but the replication never starts. But friends, whenever the CDK activity is high, now undoubtedly that a pre-replication complex can be converted into the active replication complex. And most important point every student has to remember, when the CDK activity is high, friends, remember my words, only active replication complex will form, that's all. What friends? Only active replication complex will form. What's the most important point? There will be a no formation of the pre-replicative complex. That's the most important statement. New pre-replication complex never formed, friends. Only the active replication complex will be formed. Never ever get confused with that. And the question for the exam point of view, you all people has to remember, in which phase of the cell cycle. Every student answer my question. In which phase of the cell cycle the CDK level is low? Is it a G1 phase or is it a S phase? Every one of you. In which phase the CDK level is low? Undoubtedly, the CDK level is absolutely low in the G1 phase. That's why pre-replication complex will be formed, but it never be activated. When it comes to the S phase, friends, in the S phase, the CDK level will be absolutely high. When the CDK level is high, friends, undoubtedly it leads to the active replication complex. No need to get confused with that particular point. Okay, you got that point very clearly. That is about the simple process of initiation. Now let's talk about the process of elongation. See, friends, you all people commonly know that. What do you mean by the word elongation? Elongation means synthesis of the two strands. We all people commonly know that. Leading strand, lagging strand. What else will happen, friends, in the elongation? Apart from the synthesis of a leading strand and lagging strand, in case of a elongation, proofreading also occur. Proofreading means what? Checking the errors. That also will occur. And everyone quickly recall. In case of a prokaryotic elongation, what is the name of the enzyme that is involved? You know very well, polymerase 3 is the name of the enzyme which is synthesized the leading and lagging strand in case of a prokaryotes. But all the students listen. Here, both the leading strand and lagging strand majorly synthesized by the polymerase delta only. But what is the point? The point is that polymerase delta never start the process. Because if I want to synthesize the DNA, first it is important we have to add the primer. So who is adding that particular primer? Polymerase alpha. Polymerase alpha is the one which is adding the RNA primer. So every student remember my words. What is the length of the primer? Primer is very small, 11 plus or minus 1. That's all. 11 plus or minus 1 nucleotides. Now, the moment primer got added, what is the polymerase alpha is doing? You can see that. It is only started continuing adding the nucleotides up to 20 to 50. Later, polymerase alpha can be replaced by the delta. Polymerase alpha can be replaced by the delta with the help of a pCNA. Please don't get confused. So it is in vivo, it is plus or minus 1. In vivo means within the body. But you all people know very well, when we are performing the PCR, that time also we will add the enzyme. So that time also we will add the primer. You know what is the length of that particular primer? The length of that particular primer is 60 nucleotides. Please don't get confused with that. Now, friends, I hope you all people remember. Again, I am telling you, listen my statements correctly. Actually, leading strand can be synthesized by delta. Lagging strand is also synthesized by delta. There is no doubt about it. But friends, in some cases, I am always using the word some cases only. In some cases, the lagging strand can be synthesized by polymerase epsilon instead of polymerase delta. Only some cases, not all the time. And the point is that polymerase epsilon only synthesize the lagging strand. It won't synthesize the leading strand. That is the reason, friends, we never say that polymerase epsilon is the most important enzyme. Because if it is synthesizing, it has to synthesize both leading strand and lagging strand. But unfortunately, it can synthesize only lagging strand. That's why we never say that polymerase epsilon is the major one, but polymerase delta is the major one, because it synthesizes both leading and lagging with the highest efficiency or with the highest processivity. What is the reason? Because polymerase delta is always associated with the pCNA molecule. There is no doubt about it. Now, very important statement. This is important for the exam where there is a possibility that we can expect the question. If every one of you very quickly recall my words, in case of a prokaryotic organism, who will replace the primer? Hope every student remember. DNA polymerase 1. Correct, na? DNA polymerase 1 is the one which remove the primer. And I have also told to all of you that is only replace the nucleotides also. Is it? But friends, when you look into the eukaryotic organism, every one of you listen my statements very correctly. When polymerase delta, see, remember, in case of a eukaryotic organism, polymerase delta is synthesizing all the nucleotides. When it reaches to the last, who will remove the primer? Every student remember my words. All primers, I'm using the word all primers can be replaced by an important enzyme. What is the name of that particular important enzyme? RNase H1. What do we call it as? RNase H1 is the name of the important enzyme which remove all the primers except the last one. This is the question which we got in the exam. RNase 1 which remove all the primers except the last one. Now the question is that why it can't remove all the primer? Why it can't remove last primer? When it is removing all the primers, why can't it remove the last primer? Every student, please look into the point very correctly without having a confusion. Look into this, all of you. This is a polymerase delta. Look into the point. So friends, you know, right? Polymerase enzymes always move very faster because friends, it has to synthesize millions and millions of nucleotides. So friends, it has to move very faster. Now all the students remember. So polymerase delta is moving very fast. Whenever it is moving very fast, you know what happened? Whenever it reaches to the primer, whenever it reaches to the last primer, originally friends, whenever the polymerase delta reaches the primer, it has to stop immediately. But it won't stop. Since it is coming very fast, it will come and hit the primer. What it will do? It will come and hit the primer. The moment it will come and hit the primer, you look into the picture, see this very clearly. So the primer create a flap-like structure. So this kind of structure, see, originally primer has to be like this. Now what happened to the primer? The primer got arranged like this because of that particular hitting. So this is called flap. So who is going to remove this particular flap? This particular flap can be removed by an important enzyme. We call it as FEN1 enzyme. Flap endonuclease 1 enzyme. Please don't get confused with that particular point. So finally, ligase enzyme will come and seal the nick. Always remember, all the primers can be removed by RNase H1 except the last primer. Last primer can be removed by FEN1 flap endonuclease 1. So this is a simple elongation. See, whether it is a prokaryotic replication or eukaryotic replication, friends, the process of elongation is really easy. It is not at all a difficult process, right? Now all the students listen. Whenever we talk about the termination is the very complex process. Hope every student remember in the yesterday class. In the yesterday class, we started studying the termination in prokaryotes. How does the termination occur in the prokaryotes? It's very easy, right? If everyone quickly recall, what will happen? This is called ORC, origin of replication. From here, one strand is moving. From here, one fork is moving. Both fork move at one particular point. That is called as the termination point. But in case of a prokaryotes, the DNA is a circular DNA. And you also know very well it is a very small DNA. Now, what's the most important point? The most important point is that in case of eukaryotes, the DNA is a linear DNA. It's not a circular DNA. And moreover, the length of the DNA in case of eukaryotic organism, it is a very large. And first of all, what is the problem? In case of a replication, what is the problem? Every one of you look into the picture very carefully. Try to understand this correctly. See friends, this is the origin. You know it very well. From this origin, one fork is moving in a clockwise. You know very well, another fork is moving in the anti-clockwise. There's no problem. Everyone pay attention. Listen to me very correctly. So friends, now two strands got separated and there is a synthesis of the daughter strand. Every student pay attention towards the diagram. Whenever there is a synthesis of the daughter strand, listen to me correctly. At the end, you can see, right? At the end, you can see one primer is present. Where? Towards the 5' group, one primer is here. At the same time, here also, at the 5' end, there is a presence of the another primer. Okay, you got that point very correctly. Now, friends, we have successfully removed the primers. We have successfully removed the primers. Now, the concept is very clear. Whenever we have removed the primers successfully, now the most important and interesting point all the students has to remember for the exam is that when I remove the primer, I have to replace with nucleotides. But do you think it is really possible? No, we can't replace with the nucleotides. Why? We have studied very clearly. If we want to add the nucleotides, what do we require compulsory? Compulsory, we require 3' hydroxyl group. Without 3' hydroxyl group, friends, so we can't remove it at all. Sorry, we can't replace it. Now you tell me, is there any 3' hydroxyl group is available? No. Then if there is a no 3' hydroxyl group, how does the DNA polymerase enzyme can add the nucleotide? That is not at all possible, right? As a result, friends, see here, every single generation, the DNA is becoming shorter and shorter and shorter. So friends, you also know very well, when the DNA becoming shorter and shorter and shorter, their lifespan will be decreased. And it leads to many type of tumors also. Now, what is the solution for this? Everyone, once again, first understand what is the problem. The problem is that, listen my words very carefully. The problem is that whenever the primers got removed from the 5' end, the DNA polymerase enzyme cannot replace the primers with the nucleotides. What is the reason why it can't replace? The reason is that there is no availability of the 3' hydroxyl group. What friends? There is no availability of the 3' hydroxyl group. That's why they can't add it at all. So because of no nucleotides are added, every single generation, the DNA is becoming shorter and shorter and shorter. Now, as a result, lifespan will be decreased and it leads to the cancer formation also. Now, what is the solution? Now, the solution for this is telomeres. In the DL also, they have asked the question, but they have not asked the question from here. They have asked the question from chromosomal organization about the telomeric structure. But remember, telomeres are the ends of a chromosomes. Correct, na? Now, all the students remember, you know, right? DNA contains 4 bases: adenine, thymine, guanine, cytosine. And you also know very well that the telomeres never follow Watson and Crick base pairing. This is the statement also you got in the exam. The telomeres always follow non-Watson Crick base pairing. Why? Because it did not contain equal number of purines and pyrimidines. That's an important point. Telomeres are majorly consist of guanines. Always remember that point. And very important point, friends, where this particular telomeres are present, every one of you look into the picture correctly. Every one of you focus. Telomeres are the ends of the chromosomes. You can see that. What I'm trying to explain you, telomeres are the ends of the linear chromosome. They will protect the end from the degradation. Because friends, remember my words. Suppose example, every one of you listen. Suppose example, there is a no telomere here. Every one of you listen. Suppose example, there is a no telomere here. Friends, whenever there is a no telomere here, listen my statements very correctly. Try to understand. Many degradative enzymes, many vulnerable enzymes, they will come and degrade this DNA because it is not protected. But friends, whenever we add this particular telomere, the ends of the DNA will be completely protected. Please don't get confused with that particular statement. It protect the ends, and most importantly, friends, it is going to maintain the length of the DNA. Now, all the students look into the picture correctly. What is telomere? Remember, everyone, the terminal structure of the eukaryotic DNA chromosome. That is what exactly we can call telomere. And what is the telomere is made up of? Every one of you. Telomere is exclusively made up of terminal DNA sequence and protein. Friends, please don't confuse, okay? Telomere is made up of what? It contains DNA sequence and it is also contain a protein. Very important for the exam point of view is sequence. Telomeres are rich in the T and G. But when compared to the thymine, friends, telomeres are more rich in the G only. Please don't get confused with that particular point. Look into this, friends. This is the structure of a telomere. Can you see any Watson and Crick base pairing here? So that is the statement that we got in the exam. They are not at all following the Watson and Crick base pairing. According to them, number of purines equal to number of pyrimidines. But here, can you see? Purines are always equal to pyrimidine. No. So the statement you have to remember, telomeres are always follow the non-Watson Crick base pairing. Do not get confused with that particular point. Now, what is telomerase enzyme? Remember, everyone, without having any kind of a confusion, listen to me correctly. Telomerase is the enzyme. Remember, everyone, it is made up of two things. What does the telomerase enzyme is made up of? Telomerase enzyme is made up of telomerase RNA, and it is made up of another important enzyme, very important enzyme, that is reverse transcriptase enzyme. Every student commonly know that by using reverse transcriptase enzyme from the RNA, we can easily synthesize the DNA molecule. Everyone know about that very clearly. Listen again and again. Telomerase RNA. Sorry, what the telomerase contain? Telomerase RNA and reverse transcriptase enzyme. If you look into the structure of a telomerase RNA, what is the length of this telomerase RNA, friends? The length is 150 nucleotides. Remember, and you know what is the nucleotide sequence? You can see that cytosine, adenine. And what is Y and X? Y and X means any one base. I can't tell which base. So sequence is always like this. Cytosine, any one nucleotide. Adenine, any other nucleotide. But the cytosine and adenine will be repeated in this particular telomerase RNA. Now, this is how the telomerase enzyme look like. You can see the picture very clearly, right? So inside the telomerase, what you can see, friends, you can see telomeric RNA, you can see. You got that point? Correct, na? And what else you can observe, friends? You can observe there is an enzyme called reverse transcriptase. Now, first, all the students has to understand my statement very correctly. You know how telomerase enzyme attaches to the DNA. Till today, we have never understood why the telomerase enzyme always attaches like this. Till today, we did not understand. Every student pay attention. This is called DNA. Correct, na? This is called DNA. And this is what telomerase RNA. Now you can see very clearly telomerase enzyme will come and attaches to the DNA in such a way that always it will form a overhang. Always it form a overhang. You can see this, friends. See, till here only DNA is there, right? Till here only DNA. And what about this region? This is called as overhang. We really don't know why the telomerase enzyme will attach us like this. Why it will cause an overhang? We really don't know, and still the mechanism yet to be studied. Anyway, first listen, what is our problem first? What is our problem? Our problem here, the 5' end, when we remove the primer, we can't add the nucleotides. Why? Because if the DNA polymerase enzyme wants to add the nucleotides, we require a 3' hydroxyl group. That is our problem. That's why we can't add. Now, how this problem can be solved by this telomerase enzyme? You know what is the specialty of the telomerase enzyme first? Every one of you understand. Telomerase enzyme, immediately it starts extending 3' end of the telomere. That's a very important statement, friends. What the telomere is doing, listen everyone. Telomere is not extending the 5' end. First, it is extending the 3' end. Now, the question is very, very clear. Why the telomerase enzyme is extending the 3' end? What is the reason behind that? Because, friends, whenever it extends the 3' end of the telomere, whatever it got extended, it can act like a template. I hope you are getting my point. It can act like a template. You got that point, and you also know very well. Inside the telomerase enzyme, what is present? Reverse transcriptase is present, and it is also contain RNA template. Yes or no? Of course, yes. Now, by using that particular RNA template, the reverse transcriptase enzyme can easily synthesize the DNA. So what I'm trying to explain you first telomerase enzyme extends 3' end of the telomere. Okay. The moment it extended, remember, whatever the 3' end that it got extended, friends, what it is doing, it is acting as a template. It is acting as a template for what? It is acting as a template for new Okazaki fragment. Look into that point very correctly. It is acting as a new template. So friends, now this is the enzyme. Inside the enzyme, RNA template is there. By using that particular RNA, the enzyme can easily synthesize the DNA. Simple point. But friends, what's the problem here? Anyway, we have filled the gap. We have synthesized it. There is no problem. But you see this, still there is a overhang. Still. So this is one of the reason I can say very clearly that because of this overextension also, most of the cells will undergo aging very faster, and most of the cell will become tumorized also very fast, just because of this 3' extension. So they can also give the question in the exam. What question they will give in the exam? Which of the following enzyme, which of the following DNA replication enzyme, remember my words, it makes you confused. Which of the following DNA replication enzyme involved in the cell aging? I want you to compulsory remember that is the telomerase. Telomerase play important role in the cancer cell biology and as well as in the cell aging also. Why? Because it is creating a overhang. Because that overhang become free, right? If something become free, many enzymes come and start degrading. It leads to many complications. So that's why it may cause the cells also to become very older, very fast. Don't get confused with that. So that is about what? Eukaryotic DNA replication. Okay. Is it clear, everyone? Just let me know. Clear for everyone, every one of you. Fast. Okay, that's great. Okay. So now we have to study the most important step that is called as transcription. See, friends, from now onwards, the lesson will become, little difficult. I want all the people has to understand correctly. And if you look into your 2023 set question paper, in the set question paper, they have asked 2 questions. The 2 questions they have asked from transcription only, and very easy questions they have asked. I am talking about the transcription. They just asked 2 questions And that is also very straightforward questions only they have asked. Now you look into this. What is transcription? From now onwards, every one of you pay attention and try to understand me very correctly. As all of you know very well, our ultimate goal of a DNA molecule is synthesis of the protein. This is ultimate goal. But we have studied very clearly that even though DNA is involved in the synthesis of protein, it won't involved in the synthesis of protein directly. Instead of involving in the synthesis of protein directly, every student pay attention. DNA first synthesizes an RNA molecule, and this RNA molecule only involved in the synthesis of protein. That's a very important point that I want you people to remember for the exam. First, DNA molecule can synthesize RNA, then RNA can synthesize the protein. Why? Just to protect its integrity, just to protect its stability. So now you listen. Again, most of the students will think that in order to synthesize the RNA molecule, we require entire DNA molecule. That is also completely wrong statement. Suppose this is a DNA molecule. So from the DNA molecule, especially from the template strand of the DNA, we just use a little bit of information. That's all. We don't use only a small part of the DNA. Now you can ask me, sir, DNA is a very big. DNA is a very big. Now the question is that which part of the DNA it is used to synthesize the protein? That is a good question. Which part of the DNA it is used to synthesize the protein? The part of the DNA which code for protein. The part of the DNA which is coding for a specific protein. So it depending on the protein, it depending on which protein to be synthesized, we will take that specific part of the DNA only. Don't get confused with that particular point. Now, every one of you listen. What is the name of the enzyme? And what is the process, first of all? Listen, every one of you. DNA-mediated RNA synthesis. What do we call it as? DNA-mediated RNA synthesis. We call it as the transcription. What do we call? Transcription. Now, every student remember my words. Now, the point is very, very clear, friends. If the DNA is synthesizing the RNA, that is called transcription. If DNA is getting synthesized by the RNA or from the RNA, we called reverse transcription. So without confusion, everyone remember here, I am synthesizing RNA first. From where? From DNA. Since I am synthesizing the RNA, what is the enzyme? RNA polymerase. There's no doubt. RNA polymerase enzyme only synthesize the RNA. But what is the question? The question is that RNA polymerase enzyme is dependent on what? That is the question. It is always DNA dependent RNA polymerase enzyme. What enzyme it is? DNA dependent RNA polymerase enzyme is synthesizes the RNA. Now you listen. This is the process of transcription. Now from the RNA we synthesize the DNA. So remember, I am synthesizing DNA. Since I am synthesizing the DNA, what is the enzyme? DNA polymerase enzyme. But the question is very clear. I am synthesizing the DNA from the RNA. That's why we called it as a RNA dependent DNA polymerase enzyme. This is in case of viruses, we called it as reverse transcriptase enzyme. Please don't get confused with that point. In simple words, the enzyme involved in the transcription is called as DNA dependent RNA polymerase. The enzyme involved in reverse transcription is RNA dependent DNA polymerase. Please don't get confused with that particular point. Okay, now listen. Friends, again they will ask the question, in which of the following steps of the central dogma gene expression will occur at maximum? Every student remember my words. In which of the following steps, remember everyone, gene expression occur maximum. Every student remember my words. Gene expression will start. Gene is expressing at RNA level because DNA level just replication occur. Gene expression starts at the RNA level. The point is that maximum gene expression will occur at the protein level or at the translation level. Please don't get confused with that particular point. Now all the students listen to me correctly. What is the transcription? Transcription is the first step in the gene expression. It's a very important, easy to understand and extremely important for the exam. Focus everyone. And what do we call it this as? Every one of you. So friends, transcription, every one of you, simply call it as DNA dependent RNA synthesis. As every one of you know very well, in every single organism, who is the genetic material? Undoubtedly, DNA is the genetic material, except viruses. Except viruses, all the organisms are having DNA as their genetic material. Now, every one of you, look into this line, what we have written here. Try to understand this particular line. Listen, everyone. During the process of a transcription, every student remember, an enzyme system converts. Listen, everyone. Enzyme is converting a small information. The information present in a small segment of the DNA. So this is the DNA. A small segment of the DNA converted into RNA. Now let's see how. Again, this is the question which is directly asked in your JL question paper, which is asked in the SET exam, which is a very straightforward question. And it is actually a type level of intermediate question. Okay. Now tell me, Varalakshmi, in case of a prokaryotic organism, how many number of RNA polymerases are present? Varaprasad, Murali, everyone. In case of a prokaryotic organism, how many number of RNA? Listen my question. All the students has to listen to me. No, you people are not listening my question. My question is how? No. My question, listen the question. My question is, what is the total number of RNA polymerases present in the prokaryotes? Friends, in case of prokaryotes, there is only one RNA polymerase. That one RNA polymerase only will synthesize mRNA, rRNA, and tRNA. But in case of a eukaryotic organism, friends, there are three RNA polymerases: Pol1, Pol2, and Pol3. This is in where? In case of the eukaryotic organism. Please do not get confused with the statement. Okay, now every one of you try to understand my statements without having a confusion. Listen to me correctly. You all know very well, mRNA function is what? Synthesizing the protein. tRNA is what? Bringing the information, bringing the amino acid. And what is the rRNA, which is the most versatile one among all? Among all, the most important one is the rRNA, because this is the one which is involved in the synthesis of protein. Every student keep the point in your mind. rRNA is the one which is having the highest lifespan. You know, mRNA and tRNA can degrade within minutes to hours. It will get degraded completely. But rRNA can withstand up to 2 generations. And very important statement all the students has to remember for the exam. Where rRNA get synthesized? This is only question. You know very well, rRNA is exclusively synthesized in the nucleolus. And mRNA and tRNA both can be synthesized in the nucleoplasm. And very important point for the exam, what are the enzymes involved here? All the students remember. rRNA. Remember everyone, rRNA can be synthesized by, you can expect a matching question, RNA polymerase 1. RNA polymerase 1 synthesize rRNA. mRNA can be synthesized by RNA polymerase 2. That's also a very important point. Whereas tRNA, so friends, listen. tRNA, listen my statements, all the small RNAs. I'm using a word, all small RNAs. Now you can ask me, what are the examples of small RNAs? You can take tRNA as a small RNA. You can take what we can call snRNA is also a, no, not snRNA. Sorry. tRNA and miRNA. Okay. Micro interference RNA. Okay. So these are all, and HIRNA. They are all small RNAs. All the small RNAs can be synthesized by RNA polymerase 3. Friends, please do not get confused with that. Pol1 synthesizes rRNA, Pol2 synthesizes mRNA, and Pol3 can synthesizes all the type of small RNAs. Please do not get confused with that. Okay. Now, before understanding the process of the transcription, we have to study the fundamental differences between the transcription and replication. If they ask you a very simple question, very simple, replication, transcription, translation. If they ask a question among these three steps, which is the most accurate step? You don't need to think too much. DNA replication is the most accurate step. The reason is that, friends, the transcription depends on the replication. Translation depends on the DNA. So everything is under the control of DNA, which means that when the replication is happening, friends, the replication should happen very accurately. There should not be any mistake under any circumstances. Remember that point very correctly. And if you look into the replication, all the students remember, in case of a replication, we are copying the entire genome. Yes or no? See, the genetic information is passing from one generation to the another generation. We are all looking like our mother and father. Why? The genetic information, that is the DNA, moved from their generation to our generation, which means that the entire genome got passed. Now, what about the transcription? It copies only a certain part of the genome. Never ever get confused with that particular point. And very important point for the exam, very important, you know very well. This is the DNA polymerase enzyme. DNA polymerase enzyme, if it wants to add the primer, remember everyone, it requires what enzyme? You know very well, it requires the primase enzyme. Friends, without primase enzyme, it is impossible. The DNA polymerase enzyme never synthesizes the DNA, daughter strand. And what is the specialty of a RNA polymerase enzyme? RNA polymerase enzyme itself having intrinsic helicase activity. What I am trying to explain to all of you, RNA polymerase enzyme having intrinsic helicase activity. Sorry, I'm so sorry. It is having intrinsic primase activity, not the helicase activity. And of course, friends, RNA polymerase enzyme having intrinsic helicase activity also. That statement also true. It do not require the helicase also. It will only open the double-stranded DNA very easily. Now, see the third point what I have written here. I have written a point that the product remained paired with the template. What is the meaning of that? Every one of you listen. This is the DNA, right? Listen, every one of you. Now, whenever the daughter strand got synthesized, this is the daughter strand. You can see the picture very clearly, friends. The daughter strand is remain associated with what? You can see that. The daughter strand is remain associated with the template all the time. Correct? Now, it is always bind with the parent strand. Now, but what about the mRNA? Suppose this is the DNA. When the mRNA got synthesized, friends, mRNA come out. Do you think mRNA is associated with the DNA? No. So that's why we have written a statement that product doesn't remain. Product doesn't remain base paired with the template. Please do not get confused with that particular point. And it is a less accurate process and it is a more accurate process. Please don't get confused with that point. And friends, I can say one of the major reasons why the DNA replication is more accurate is, according to the recent research, the versatile DNA polymerase enzyme will make just one mistake, single mistake out of 10 power 6 to 10 power 7 nucleotides added. Which means, friends, out of 10 lakhs to 1 crore nucleotides that it is added, it just make one mistake. So that much accurate enzyme it is. And very interesting point is that, friends, Even though it made a single mistake, what's the point? That particular single mistake also can be repaired by using DNA repair mechanisms. We will see it. But if you look into the RNA polymerase enzyme, it just make one mistake out of every 10,000 nucleotides added. It doesn't mean that it is not an accurate process. It is accurate, but compared to the DNA replication, friends, it is less accurate. That is what I'm trying to explain you. Okay, now you remember whether it is a replication, transcription, translation, what I told to all of you, these three steps are common. Initiation is common, elongation is common, termination is common in any organism. But friends, before starting the process of the transcription, we all people has to understand what are required for a transcription. Simple example, if you want to prepare a dish, if you want to prepare a healthy dish, first you require all the vegetables or whatever is required. First, you have to arrange all. Once you arrange all, then only you can prepare a dish. Similarly, if you want to start the process of transcription, first we require a machinery. What are those? RNA polymerase enzyme and promoter sequence. I can tell you in a simple words, this is a DNA molecule, correct? Na? And this is an RNA polymerase enzyme. The question is very clear. If the RNA polymerase enzyme want to synthesize the mRNA, RNA polymerase enzyme has to attach us to the DNA. Now, question is very, very clear. This is a very big DNA. How does where the RNA polymerase attaches to the DNA? RNA polymerase having a specific sequences on the DNA. A specific sequences on the DNA. The area or the sequence where they will come, where the polymerase will come and attaches, that is called promoter. Now the question is crystal clear. This is RNA polymerase enzyme. You understand that RNA polymerase enzyme attaches to the promoter. Now the question is very clear. How RNA polymerase comes to know? How RNA polymerase comes to know where is the promoter? Who is guiding? How it will find it? RNA polymerase enzyme is guided by transcription factors. They are the one which required for initiation, required for elongation, which required for termination. Don't get confused with that. And what else we needed? We require activators. We require repressors. What is activator? Activator is the one which will activate the process of transcription. Repressors are the one, name itself indicating, right, which repress the process of transcription. Never get confused with that particular point. Okay, now let's talk about it. Again, the most commonly asked question, every one of you remember, polycistronic and monocistronic. This is a very important one. Poly, many, cistronic, group of genes. In single word, I can tell you in a single line, one mRNA giving rise to many proteins. One mRNA giving rise to many proteins at a time. And what about the monocistronic? One mRNA, one protein. One mRNA, one protein. And the point is very clear. We, eukaryotic organism, we, human beings, plant, yeast, they are very complex organisms, right? So these complex organisms always follow monocistronic. One mRNA giving rise to one protein. My example, friends, this is a mRNA which is going to give rise to the hemoglobin in our body. Now tell me, this mRNA give rise to the myoglobin in our body? No. Myoglobin can be synthesized by another protein. Do you think this mRNA can synthesize collagen? No. Collagen can be synthesized by another mRNA. So what I'm trying to explain you, one mRNA give rise to one specific protein. Do not get confused with that. But if you look into the prokaryotes, one mRNA, multiple proteins. Anyway, we will study in the operons, but you all people know, right? In case of a lack operon, lack Z gene, lack Y gene, lack A gene, all can be synthesized from one single mRNA. Correct, na? That is what we call it as polycistronic and monocistronic. Now, friends, now let's try to understand how does RNA look like? How does RNA look like? Whether it is a prokaryotic organism or whether it is a eukaryotic organism, how does the mRNA look like? Every student look into this. Now listen, I will tell you. Again, some students will get a doubt that does the length of every single mRNA is the same? That's also a very good question. Does the length of every single mRNA is same? No. Remember, every one of you, this is an mRNA. Every student commonly know that mRNA can be synthesized in 5' to 3' direction. You all people know about this very well. Now, you also know very well, mRNA means what, friends? mRNA, messenger RNA, which means that it is carrying the information. It is bringing the information. From where it is bringing the information? From the DNA. And you know very well how it will bring the information. It will bring the information in the form of codons. Never ever get confused with that point. Now you all people know very well. Some students will think that entire mRNA can code for the protein. That is absolutely wrong. Please don't think that entire mRNA can code for the protein. No. Listen, all of you. On the mRNA, there are certain regions are there which never code for protein. I'm not talking about the exons and introns. Okay, forgot about that. See, listen, everyone. So at the 5' region, there is at the 5' end of the mRNA, there is a sequence we called UTR sequence. What is the full form of UTR? Untranslated region, the region which never undergo the protein synthesis. The UTR present in the 5' direction, friends, we called it as leader sequence. Never get confused with that particular point, right? And at the same time, friends, at the 3' end also, there is another UTR region is present. And this UTR region, we generally called it as trailer region. What do we can call? Trailer sequence. One is leader, one is the trailer sequence. Please don't get confused with that particular point. And friends, you all know very well, the mRNA starts with a start codon called as AUG. Then many codons are present. Now the question is here, the point is here. The length of the mRNA is depends on how many number of codons are present. What's the question, friends? The length of the mRNA depends on how many number of codons are present. Now you can ask me a question, sir, how do we know how many number of codons are present? Renuka, tell me, Mahip already tell me, if I want to synthesize a protein called as insulin, how many number of codons I require? Every one of you. If I want to synthesize insulin, how many number of codons I require? Aishwarya? See, you all people know, right? Insulin is made up of how many amino acids? Very good. If I ask directly insulin, right? If I ask a pro-insulin will be 84. I ask directly mature insulin is made up of 51 amino acids, which means that one amino acid is coded by one codon. Correct, na? So friends, in order to synthesize the insulin, how many codons we require? We require 51 codons. So it is very simple. So the mRNA also contains 51 codons. Now all the students, listen, example I'm telling you. Now, I hope you all people remember. Every one of you answer. An enzyme called as pyruvate kinase enzyme. Everyone tell me, the pyruvate kinase enzyme, please turn off your video. Okay. Now my question is, where this particular pyruvate kinase enzyme is present, where you studied? Every one of you, in which reaction you have studied the pyruvate kinase enzyme? In which step? Forgot everyone. Pyruvate kinase enzyme, every one of you. No, I am asking in which, very good, very good, Maheshwari. In the glycolysis 10th step we have studied, right? Now you know, pyruvate kinase enzyme is nearly made up of 20,000 of amino acids. I hope every student is understanding my statement. And if you take an example of a urease enzyme, it is made up of 50,000 amino acid. So friends, you people try to understand what I'm trying to explain to all of you. How big the mRNA is depends on what kind of protein that it is synthesizing. Some proteins are very small, mRNA is also very small. Some proteins are very large, mRNA is also extremely larger in size. Now, at the end of all the codons, it contains 3 stop codons. Every one of you commonly know that this is also a direct question which is asked in your exam. UAA, UAG, UGA. Correct, na? Amber, Walker, Opal, based on the name of the discoverers. Amber, Walker, Opal. They do not code for any kind of a protein synthesis. But I strongly believe you all people remember one question. Every student answer now. What is the name of the amino acid that is coded by the UGA? I want at least few people has to answer this question. Tell me, Maheshwari, what is the name of the amino acid that is coded by the UGA during the time of a post-translational modifications? Anyone, anyone? Very, very good. Very good. What is that? No. Who said that valine and glycine? Huh? Valine and glycine are basically coded by normal amino acids only, normal codons. I'm asking here, UGA is the stop codon, right? UGA is actually coding for an amino acid called selenocysteine, only which is added during the post-translational modifications. I'll explain you in the translation. Remember that. Valine is one of the 20 amino acid, right? Glycine is also one of the 20 amino acid. My point is very clear here. I'm just saying to all of you, there is a stop codon that can code for one amino acid during the post-translational modification, which is called selenocysteine. Remember the point correctly. Okay, now you understand what is polycistronic. You understand what is monocistronic also. Now look into the point very correctly. What is promoter? What is the definition I have given to all of you? The promoter means promoter is a nucleotide sequences. Promoter nothing but what? They are the nucleotide sequences only, right? They are the group of nucleotide sequences which are acting as a binding sites for the versatile enzyme RNA polymerase. RNA polymerase is the enzyme which will come and attaches to the specific sequences that are present in the DNA. That binding sequences only we can call the promoter. Every student keep the point in your mind. This is also they will ask the question directly in the exam. Listen, every one of you, very carefully what exactly I'm trying to explain you. Remember, everyone, this is the gene, nothing but DNA. Remember all of you. Remember all of you. This is what? This is the point. This is the point where the transcription will start. Hope every student remember, in case of a replication, where the replication will start, it will start at a point called as ORC. Similarly, the point at which the transcription will start, that particular point only we people generally called as INR, initiation region, where the process of transcription will start. Now, what some students will think? Students will think that RNA polymerase enzyme directly comes and attaches to the INR region and they will start the synthesis of mRNA immediately. That statement is absolutely wrong statement. Remember, first RNA polymerase attaches to the promoters first. Once it attaches to the promoter, from the promoter, they start moving. Where? They start moving to the INR. Now, the question is crystal clear. In case of a prokaryotic organism such as E. coli, friends, where this particular promoters are present. Every student listen. The promoters are present left-hand side. You can see that. So this is the INR. Towards the left side of the INR, promoters are present. But remember my words. But if you look into a complex organisms like us, in case of humans, the promoters are present left-hand stream also, right-hand stream also. Widely we can call it upstream, downstream. But in case of prokaryotes, friends, it is exclusively present only in the upstream. Never ever get confused with that particular point. How many promoters are there? Friends, in case of eukaryotes, you can find multiple primers, many and many primers. And but in case of a eukaryotic organisms, friends, remember my words, there are only two promoters are there. One promoter located at -10 region, another promoter is located at -35 region. So why did I use the word minus? What is this 10? What is this 35? Friends, from the INR, if you keep counting the nucleotides, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, at -10, it is a left-hand side, right? That's why we can call -10. And again from here, -35. So there will be a presence of the presence of the promoters. Their RNA polymerase will come and attaches. You got that point? So in case of a bacteria, there are only two promoters are present. One is called -10 and another one is called -35. Please don't get confused with that particular point. Now, friends, that -10 region we can call Tata box, TATAAT, because it is having a repeated sequence of thymine and adenine. And sometimes instead of using the word Tata box, they will use the word Pribno box. Anything is same, friends. Pribno is the name of the scientist. Pribno is the name of the scientist who identified that particular region. And there is another sequence called -35 contain TTGACA. I don't encourage all of you to remember this because they won't ask you. I want you to understand where the promoters are present. But friends, 90% in all the prokaryotic organism, only these two sequences are present. Only these two promoters are present. -10 and -35. But recently, scientists identified that in some organisms, there is another promoter is also present. You know where it is located? -40 and -60 region, there is another promoter is located, which is rich of adenine and thymine. Every student, please look into the diagram very correctly. No need to get confused. It's very easy to understand. Look into this. This is the point. This is the area where RNA synthesis is starting. Every student look into this. I am talking about, very good, Richard and Gauss. The organism you can take type of bacteria only, Balakrishna. I am not very sure about which is the specific bacteria. Okay, which is specific bacteria I cannot say, but you can see this. I have written here a bacteria which contain a gene named as RRNBP1 gene. In that particular gene, these sequences are present, and it's not required to remember the organism. So this is the RNA transcription start site. Look into this. -10 nucleotides. That's why left-hand side, right? That's why -10, -35. At -10 Tata box, at -35 TTGA sequences are present. Okay, you got that very, very clearly. They are, you understand the concept correctly. Now the question is very clear. There are two type of promoters are present. What are the two type of promoters? Strong promoter, weak promoter. And again, some students will get weak promoter means won't it involved in the transcription? It is not like that. Okay. Whether a promoter is a strong promoter or weak promoter, RNA polymerase compulsory will attach us. Transcription factors will compulsory attach us. There is no doubt about it. Now the question is that why did we use the word strong promoter? Because friends, the promoters which is having the sequences very close to the consensus. Friends, remember, consensus you can also call it as a conserved sequences. You can also call it as repeated sequences. Example, TA, 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 TA. What is this? It is completely repeated sequence. This is called conserved, consensus, repeated. So if a promoter contains this kind of sequence, it is a strong promoter. What is weak promoter? Remember everyone, weak promoter means which is having a less consensus sequence. Means, example, TGCA, AT. This is not highly repeated like this, right? That's why we can call it a less consensus sequence. Please don't get confused with the point. Now, all the students listen. Now, this is the question which is asked in your exam in the 2023 set question paper also. You got the same question from the enzyme. I hope you all people remember, right? in the replication only we have seen the structure, no? If everyone quickly recall, RNA polymerase and DNA polymerase, what they having, friends? Either magnesium or zinc. How many aspartate residues are there? 3 aspartate residues are present. That is a simplified structure. And I have also told to all of you very clearly that if the RNA polymerase enzyme wants to be super active, it compulsory require the guidance. It compulsory require the information from the DNA. That's why we call it as DNA dependent RNA polymerase. Every student look into the structure first of all. So this is the direct question which you got in the exam. RNA polymerase enzyme. Everyone tell me quickly, how many number of subunits are present in the DNA polymerase 3? Every one of you. How many number of subunits are present in the prokaryotic DNA polymerase? Forgot everyone. My question is, how many number of subunits? Very good. There are 10 subunits. How come you forgot? 2 days only now we discussed. 10 subunits are present. Correct, na? Similarly, friends, how many number of subunits are present in the RNA polymerase enzyme? I am talking about only prokaryotic RNA polymerase, friends. I am not talking about eukaryotic RNA polymerase. It generally consists of 6 subunits. No, it's not 5. It contains 6 subunits. 2 alpha units, 1 beta prime, and 1 omega. So everyone, whatever you said is right. Whatever you said is right. Remember, this is called core. This is called core enzyme. So you can ask me a question, sir, what is the meaning of a core enzyme? Every student use your brain. Core enzyme means the enzyme which is involved in the synthesis of RNA. The enzymes which are involved in the RNA synthesis, that is called core enzyme. But what is the interesting point? All the students listen. This is the DNA. This is the RNA polymerase enzyme, RNA polymerase core enzyme. This is the promoter. Now every student know very well the core enzyme has to come and attaches to the promoter. Now the question is crystal clear. How does the core enzyme comes to know? Where is the promoter? So someone has to guide the core enzyme. Who is guiding the core enzyme? A sigma factor. What was that? Sigma factor is the one will guide the core enzyme to come and associated with the promoter. They can ask a question, which part of the RNA polymerase enzyme never involved in the transcription? Never involved means I am talking about which is not completely involved in the initiation, elongation, termination. That is sigma factor. Now, what's the function? Now, ultimate point you have to remember, sigma factor is exclusively responsible for recognizing the promoter. So, friends, remember every one of you, core enzyme means having 5 subunits. This entire green color you can see, right? That is called core enzyme. Whenever the core enzyme associated with the sigma factor, the blue color one, then only, friends, we call the enzyme as holo enzyme. What is the meaning of a holo enzyme? Holo means complete enzyme. When you can call an enzyme is a complete enzyme when it contains core subunit plus sigma factor. Never get confused. Now you look into this beautiful picture. All of you pay attention. Look into this. This is an RNA polymerase enzyme. Correct, na? Now listen. This is an RNA polymerase enzyme. I did the cross section. You can see this. I did the cross section of the RNA polymerase enzyme. When I did the cross section of a RNA polymerase enzyme, look into this. How many strands, how many channels it contains? It contains template strand channel. You can see that. And this is called non-template strand channel because we require only one strand, right? And first of all, how does the RNA enter into the DNA? How does DNA pass through the polymerase enzyme? There is a channel called as downstream channel. Now, friends, you know very well. If I ask you what is the meaning of a synthesis of DNA or synthesis of RNA, what you will tell? You will tell that addition of one nucleotide with another nucleotide only called as synthesis. Correct, na? So in which direction nucleotides will be added? You can see this. Nucleotide triphosphate uptake channel. Like this, nucleotides will be added. And RNA comes out in this direction. That's why it is called RNA exit channel. Exit channel, nucleotides adding channel, template strand, non-template strand. Now all the students has to understand. Most of the people having a confusion here. What do you mean by the template strand? What is meant by non-coding strand? What is sense strand? What is anti-sense strand? What is non-sense strand? Every one of you try to understand what exactly I'm saying here. Listen to me correctly. This is a mRNA, correct? Na? mRNA is in which direction? Every student know that mRNA always synthesizes in the 5' to 3' direction. Okay. Now tell me, all the students tell me, tRNA is in which direction? Aishwarya, Vishwanath, tell me, tRNA is in which direction? Every one of you. Balraj, tRNA is Maheshwari, in which direction? tRNA synthesized in which direction? Of course, friends, tRNA is also synthesized in the 5' to 3' only. You forgot it. Where the amino acid is added. Remember everyone, amino acid is added at the 3' end of the tRNA. Is it or not? So the tRNA is also synthesized in the 5' to 3' only. That we'll discuss in the process of translation, don't worry. Okay, now listen to me. Now listen to me. Look into the point. This is the mRNA, right? So mRNA is synthesizing in the 5' to 3'. Now the question is very clear. This is our DNA molecule, right? If I want to synthesize the DNA molecule, every student first listen to me. Very carefully understand. If I want to synthesize the DNA molecule, if I want to synthesize the RNA, I require one strand of the DNA. Which strand? Why should I take only that strand? Remember everyone, I always require a strand. Listen my statement correctly. I always require a strand which is complementary. Whichever the strand is complementary, that is called as template. You look into this. So remember everyone, you can see this. G pair with C, C pair with G, G pair with C, A pair with U, T pair with A pair with U. My friends, what do we call this as? Complementary strand. So that complementary strand only we can call the template strand. Now the question is very clear. Now you have to ask me, sir, why can't you take this strand? You tell me, how can I take this strand? The information is same. CC, GG, CC, TU, AA, TU, AA. Information is same. If the information is same, how these two can be complementary with each other? How can I synthesize mRNA? That is not possible, right? So I won't take this. Whichever is not involved in the RNA synthesis, we generally call it as non-template strand. Whichever is involved in the RNA synthesis, that is called template strand. Now all the students, this is easy to understand. Now where most of the students get confused? Most of the students get confused. Listen my statements. I am calling it as non-template strand. But friends, it contains information. Yes or no? It contains a lot of information. See, CGC is a codon, TAT is a codon, AGC is a codon. It contains codon, it contains information. The only point is that it contains information, but it is not involved in the protein synthesis. That's the reason, my friends, we call it as a non-template strand, but we call it as a coding strand, which means it code. It contains an information. So it contains information means it makes some sense. We can call a particular person, that person is not using any sense, means he's not thinking anything. So, but here it contains information. So I can call it as sense strand. So it contains information, it makes some sense, but it is not involved in the RNA synthesis. That's why we can call non-template. The point is here, this is the DNA template strand, which is complementary. It is always complementary. You remember this point. It is not make any sense. It just act like a complementary strand. That's all. So template strand, you can call non-coding strand. So again, most of the students will get confused. Sir, why you are using a word non-coding? If it is a non-coding, how it can synthesize the RNA? You people are not understanding. Our ultimate goal is to synthesize the protein. If I want to synthesize the protein, remember every student, if I want to synthesize the protein, I require the codons of mRNA. I hope you are getting my words correctly. But if I want to synthesize the mRNA, first I require a strand. So that strand is only a complementary strand, but it is a non-coding strand. And it is a non-coding, it doesn't make any sense. So I can call it anti-sense strand. Sometimes I can call it as a non-sense strand. So this many names are there. It's very important to remember all the names. DNA template strand, or complementary strand, or non-coding strand, or antisense strand, or nonsense strand. And this is called coding strand, non-template strand, sense strand. Always remember the points. Do not get confused with that particular points. Okay, now listen. And I don't know if anyone observed it or not. In DL zoology question paper, this is the question was appeared. Right. And in the previous set also, you got this question, I think in the 2019 paper. Now remember my words correctly. What you all people said, sigma factor is the one which is responsible for helping the RNA polymerase enzyme to recognize the promoter. Absolutely true. Now please remember, there are many sigma factors are there, friends. Sigma factor 70, sigma factor 30, sigma factor 45. There are many sigma factors are there. But friends, the major sigma factor that is involved in the transcription is the sigma 70. Look into the point. So you can see this. What was this? This is RNA polymerase enzyme, which is the core enzyme having 5 subunits. And this is the sigma factor. If you look into the structure of the sigma factor very correctly, friends, sigma factor having 5 subunits. Sigma, sigma 2, sigma 4, and sigma 3 will be located here. Sigma 3 do not have a much function. Now, this is the most interesting and very important point. Listen, every one of you correctly. RNA polymerase enzyme has to recognize the promoter with the help of this. Now, question is very clear. I told you RNA polymerase recognize the promoter with the help of sigma factor. This is correct. Now my question is, how does the sigma factor comes to know where is the promoter? That's also a very good point, right? How does the sigma factor comes to know where is the promoter? Friends, again, with the help of a certain binding sites. All the students listen to me correctly. This is called sigma 4. This is called sigma 2. Every student remember, this is sigma 2 region having some aromatic amino acid. Every student answer my question. What is the example of aromatic amino acid? Give me the example of an aromatic amino acid. Every one of you. Aishwarya, what is the example of an aromatic amino acid? Aromatic. Okay. Everyone. Okay, what about others? What happens to others? Everyone forgot it, huh? All the botany people answer. What is the name of the aromatic amino acid that is involved in the auxin biosynthesis? Every one of you. What's the name of the aromatic amino acid that is involved in the auxin biosynthesis? That is the tryptophan, right? So, phenylalanine, tryptophan, correct, na? And tyrosine. These three are the aromatic amino acid. Correct, na? No. We should not say benzene ring, Balraj. We should say indole ring. It contains indole ring. Of course, whatever you said is right. Right. Now listen to me. The sigma 2 having some amino acids. Now with the help of that aromatic amino acids, sigma 2 will come and attaches to the -10 region. And how does the sigma 4 attaches to the -35? With the help of a helix, turn helix. You know, what do you mean by that? Sigma 4 attaches to once, it is called helix. Then it will turn again helix. It will turn again helix. It will turn again helix. This is called helix turn helix. This is called a very tighter attachment. Please don't get confused with that particular point. I want all of you has to understand this properly without having any kind of a confusion. Now, very important statement. We all people studied that whenever the DNA polymerase enzyme, whenever the helicase enzyme is moving very faster, it create a lot of coiling, and because of the coiling, DNA undergo the tension. Then the topoisomerase enzyme will come and remove the coiling. But you all people has to remember, I told you, right? I will tell you in the upcoming slides. RNA polymerase enzyme itself act like a helicase. Friends, remember, just now we studied, right? This is the double-stranded DNA, but we require only one strand. If we require only one strand, compulsory we have to separate the two strands, yes or no? If I want to separate the two strands, I have to break the hydrogen bonds. Who will break the hydrogen bonds? Helicase is coming? No. RNA polymerase itself having the helicase activity. It is only breaking. Now again, whenever it is breaking, it creates the tension, no doubt about it. Then again, topoisomerase enzyme comes into the picture and remove the coiling. So they can ask a very simple question in the exam. And this is the question also asked in many exams. The question is what? Most of the students will get confused. The question is, which is the most common enzyme? Which is the most common enzyme in the replication and transcription? 90% of the students will think that why there is a common similarity, because replication is different, transcription is different. So remember, the common enzyme is this particular topoisomerase involved in removing the coiling. I can't say that the helicase is involved. I can't say that. Why? Because RNA polymerase itself having helicase activity. I can't say that the primase is involved. Actually, in the transcription, there is no requirement of the primase only. But RNA polymerase enzyme itself having primase activity. So the only common enzyme in the replication and transcription is topoisomerase. Don't get confused with that particular point. Okay, now all the students remember. So far, whatever we have studied is the steps. Now what we have to understand is the stages. Stages involved. That is called initiation, elongation, then followed by, friends, termination. This is very easy. I will complete in the tomorrow class. Okay. So since tomorrow is a Sunday, I want every one of you to attend compulsory, because the only time I am having is 11 to 11:30. I mean to say, I have a free time. I will start the class either 11 or 11:30 a.m. Okay. I'll take 1.5 hours to 2 hours. I'll complete it. Again, genetics we will start in the morning only in the Monday. Okay. 4:30 a.m. onwards. Maximum in by 9th, I will finish both units, molecular biology and genetics. Okay. I'll inform the timings early morning, either 11 or 11:30. Compulsory attend everyone. I will complete the up to eukaryotic transcription. If we complete eukaryotic transcription, we can finish the translation very fast. Okay. Clear everyone? Attend compulsory. Don't neglect it. Okay. Okay. Perfect. If anyone having any doubts, you can ask. Yeah, tell me, Aishwarya. Quick. Aishwarya, I'll inform, because I have other class also, no? Based on the completion of the class, I will let you know. Okay. 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 Perfect. Perfect. Okay. Thank you all. Thank you.